Good morning, students. Today's class, this is for not only for the 4th sem B Pharmacy students, it will be more useful for even for the Pharm D students and even D Pharm students, because it is about the cultivation and collection of crude drugs, medicinal plants. So it's a chapter for every syllabus. So I have taken commonly, not only that, specifically they are asking more question for 4th semester students. They used to ask 10 mark question, 5 mark question, 2 mark question. Why not this class is useful for even for writing government exams with MCQs? Because I have taken in such a way, not very detailed, but in a brief manner, with definitions and with some of the important headings and details about them in a manner which can be easy for you to write the exam. So today we are going to discuss about in cultivation collection, apart from that wild plant collection and the comparison with the cultivated plant collection of medicinal plants. Plant variation, this is one type of question which comes to everyone. Even though the same species is grown in the same place in different cultivator, it gives different constituents, different sizes, and different characters have been observed. So that's why this plant variation? How? So we have made into 3 categories for this plant variation, in which first one is environmental, exogenous factors we are going to discuss, and endogenous factor like gene factors. And then after collecting the drugs processing, which are also having some influence in plant variation in their constituents and quality. So this is what the 3 things we are discussing, but in the 3 things there are many points. For example, exogenous factor, environmental factor, we are going to discuss about effect of climate, temperature, rainfall, day length, radiation, latitude and altitude, soil conditions, collection section, developmental age of herb, herb condition and parasites which are growing together, and dealing with same thing environmental, in that use of growth promoting substances, plant growth hormones, different hormones and their chemical structure, we will discuss about that, or plant growth regulator, because some of them growth inhibitor also there, growth regulators also there, cell dividing hormones are there. So we will discuss about that. That's why plant growth regulators. And we should know about what is allelopathy, how it is having influence with the plant growth, and in allelopathy what is symbiosis and what is antibiosis. So these all things we are going to discuss today. And also the second factor which is affecting in the variation of the plants while cultivating and collecting them, growing them in different atmosphere, we can see that endogenous factor, that is genetic factor, genetic differences we are going to see, polyploidy, plant breeding methods, in that selective breeding, hybridization, transgenic, medicinal plants. And the third factor for the variation of the plants in their production and in their cultivation, the third one is post-cultivation factors. So, this is the three things which students should remember. Exogenous factors, endogenous factors, and post-cultivation factors, in which harvesting of herbs, preparation of herbs, drying of crude drugs, adulteration of crude drugs, in which adulteration we have had a separate chapter, but still some of the definitions, some of the things which you should know, that because it is post cultivation factor which has variation in the plants which will show the factor. So adulteration of crude drugs, in that inferiority or inferior drugs, spoilage drugs or spoilage, admixture, sophistication, deterioration, substitution. So these all comes under in this post cultivation factors which will have the variation in the plants. And genetic endogenous factors, and we are going to see the exogenous factors like climate, temperature, and with the hormones also we are going to see. Now the first one, starting with the cultivation, collection, preparation of crude drugs. Students, all the pharmacy students of 4th sem B pharmacy, Pharm D students, 2nd Pharm D who is studying about pharmacognosy and pharmaceuticals, and pharmacognosy who is studying D Pharm students, please try to listen and elaborate it by accordingly by referring books. If you are not subscribed my channel, please subscribe so that it will be easy for me to give my lecture with my interest, and it will come a support from your side. Thank you. Anyhow, we will start with the crude drugs, cultivated or uncultivated plants. Some cases pharmacopoeias specify cultivated species such as fennel, ginger, cinnamomum, and opium. This is fennel, this is ginger. Opium, only officially with permission growers may legally produce herbs. You know that opium, this is an exudate latex which is obtained from the fruits by making insertion. It's an unorganized drug. And other herbs like senna, tragaganth, this is senna, tragaganth, a gummy exudate obtained from Astragalus gummifer, may be collected from wild or cultivated species. So here the question comes, what's the difference between the cultivated and wild one? So disadvantages of wild collection. There are many disadvantages when you are collecting from wild. So that's why we are talking about the cultivation and collection which helps in pure one. So we can see sparse distribution, very rare plants distribution like Scletrium totusum. It's a plant which is having more useful in various mood elevators. Anxiety patients can be treated in Africa. It is very common South Africa plant. And sparse means few or rare or less, the meaning. Potential differences to transport herb to areas or area to post-processing. From wild it will be difficult. Difficult access, forest, mountain, etc. Collectors' ignorance, admixture of other plants, collection of undesired plants parts in the original or stage of development or during an incorrect season sometimes collecting, loss of medicinal activity when you are collecting in wild, damage of natural environment, extinction of a species it can be seen. This type of the picture which is shown is a very rare species of very distribution of plants. Like extinction means occurs when species are diminished because of environmental forces, habitat fragmentation, global change, natural disaster, over exploitation of species for human use, or because of evolutionary changes in their members, genetic inbreeding, poor reproduction, decline in the population number. These all things happens. Succulent herb which comes in this, when you say this herb, Scletium tortuosum is a succulent herb commonly found in South Africa, which is also known as canna, which literally means chewable things. Sometimes it is chewed by the people, local people. It has also been snuffed and also smoked. This is the developed one. It's available in the herbal formulation powder. It is possible that Scletium may cause elevated mood and decreases anxiety. It can be treated. Stress and tension can also be treated. And we are talking about the cultivation of the plant in organic farming of medicinal plants. You might have heard advantages of cultivated herbs. We have seen the disadvantages of the wild collection. Only desired species are collected. Uniform quality can be seen. Collection, transport and access to processing facilities is improved. Better control of soil quality, pest and plant disease. Supply, constantly and regular control can be seen. Herb collectors will be trained. Plant variation. Now we are talking about plant variation. Even though collected or wild, there is plant variation. So that's what the 3 points which we are have to discuss. Ideally, collected cultivation and harvesting is aimed at producing for what? High quality, healthy herbs, complicated all natural products, but still variation between individual plants in plant size, content of the medicinal activity. Why brings about this variation? This is the question. And we say variation factors are there. There are 3 factors mainly. Makes the variation in the plant size, content of medical activities. Variation in the individual plants can be seen by means of 3 groups. That is, group of definitions are there. That is environmental, exogenous factors, genetic, endogenous factors, post cultivation factors like drying, storage methods, etc. So 3 points, we are going to see all the details. But environmental, exogenous factors, there will be many. Genetic, endogenous factors, more critical, and there are few. And post cultivation processing, drying, storing methods. These 3 groups. First one, plant variation factor number 1, which is affecting the variation of size and constituents, and the macroscopy, microscopy having some variation. Why? environmental exogenous factors. This one we can see exogenous factors in which climate, altitude and latitude, collection, season, soil condition, developmental stage of the plant, plant condition, use of growth promoting substances, allelopathy, parasites, these all things. The first one is effect of climate. Plants should be cultivated in conditions which are similar to the plant's natural habitat. Most herbs grow best in sunny, dry conditions. Factors affecting climate, temperature, rainfall, day length. These all are affecting the plant's development and their constituents. Temperature, major component. After both growth, development and metabolism of the plant, each plant is specialized to adapt to its native environment, but most are to exist in wide temperature ranges. Example, tropical and subtropical plants in temperate regions. We want to tell about Datura stromatonium. Temperature affects plants' chemical reaction rates. Example, Datura stromatonium, low alkaloids in cloudy, rainy weather, winter season. Example, volatile oils are produced more readily in warmer weather. Yet, very hot days lead to a physical loss of oil. Growing peppermint in shade rather than the sun. Rainfall. Consider annual rainfall distribution, humidity effect, soil type may influence the production of glandular hairs. Glandular hairs as shown in the picture. Continuous rainfall, loss, leaching of water, soluble substances through leaves and roots, glycosides, tannins, flavonoids, some volatile oils also can be washed off. Leaching can take place. Day length, amount and intensity of light needed differs from herb to herb. Affects the amount of glycoside, alkaloids and volatile oil produced. Example, peppermint. We know very well this plant, that's why peppermint, also known as mentha, Blasphemia wild, is a hybrid mint, a cross between watermint and spearmint. Long days, that is lengthy days, menthol, which is the constituent of peppermint, menthoferon, traces. Short days, menthoferon, main component. Other species produces more active constituents at night. Example, Nicotiana. Nicotiana tabacum or cultivated tobacco is an annually grown herbaceous plant. Plants have a variety of developmental, physiological and growth responses to light, sometimes only a particular wavelength of light. a phototropism. A plant bends or grows directly to the response of the light. You can see the picture. The shoot system bends towards the light in the picture. That is sunlight, straight it goes. But when you see the root, opposite side it goes. The root, opposite sides it goes. That is phototropism. Shoot usually moves towards the light. Root usually moves away from it. The photoperiodism, flowering, and other development process are regulated in response to the photoperiod or day length. You see this? The yellowish, round in the yellowish says that day. There are flowers which are having short day plants and long day plants. In short day, they are flowering in that particular darkened shade in the graph. And long day plants, it needs the shade or the darkness. In a particular period of time they will flower. So short day plants flowers when day length is below a certain threshold. The threshold, the word, plays the role. While long day plants flower when day length is above a certain threshold. So the picture shows very clearly. So you should know this word of photoperiod or day length. Two of them, that is phototropism and photoperiodism. One is flowering, another is about the shoot system and the root system, how they towards the development or towards the light. Radiation. Type of radiation plant receive. Example, Osimum basilicum. We know the Tulsi, Rama Tulsi, Krishna Tulsi, which is very common, which we are using in our places. Plant grows in glass house, have less phenol and terpenoids in the leaf, flavonoids and volatile oils. This is the Tulsi, Rama Tulsi. Ultraviolet radiation affects the plant's growth and development in many ways. First, it gradually stops seed growth and sprouting. Dependent on the how much radiation is released, soil can become compacted and lose the nutrients needed for plants to grow. Example, tomato. You can see this picture, the three different types of plant grown. In high UV light, it is short. Low UV light, little more higher. And UV light was protected, it is very high. So that's what we have shown. This is the picture of tomatoes protected from the UV light. Osimum basilicum is there, Rama Tulasi. Family Lamiaceae. Basil in Europe is an herb. The parts of the plant that grow above the ground shoot system are used to make medicine. Basil is used for stomach spasms, loss of appetite, intestinal gas, kidney conditions, fluid retention, head cold, warts and worm infection. It is also used in treat snake and insect bites. Latitude and altitude. Gentian bitter constituents increases with altitude. Gentian barks, which you can see, that is benzyl rhizomes. Time constituent increase with altitude. Time constituents are increased by the height of the altitude. Other oil producing plants reach a maximum at high altitudes. Like peppermint, altitude can impact essential oil composition. Plants grow at high altitudes often experience greater environmental stress and leading to the production of more potent oils. For instance, peppermint grown at higher elevation tends to have higher menthol content, enhancing its therapeutic benefits. Fat soil production may be influenced by latitude. Peanuts, olive trees grow in subtropic areas have a higher unsaturated fatty or fat content. So latitude and altitude we have briefly explained. In the exogenous factors which are affecting the plant variation in cultivation. Collection season also, active constituents of herbs are affected by the seasons due to climate, rainfall, day length, etc. Medicinal plants should be therefore collected in the season in which their active constituents are highest. Rhubarb is a general term used for the cultivated plants in the genus Rheum. Emodium. Rheum is the family in the family Polygonaceae. Example, rhubarb laxative. It is a laxative drug. Contains high anthranol in winter. Alcoholic groups which will be converted, it gets oxidized and becomes anthraquinone in summer. Soil condition, soil character, composition, permeability, porosity, all affects the health of medicinal herbs. Example, chalky soil, poor digitalis growth, cardiac glycoside. Digitalis is a genus of 20 species in herbaceous perennial shrubs, and biennial, commonly called as foxgloves. Family is Crophyllaceae. Nutrient content of soil crop rotation, nitrogen, N2 nitrates, phosphate, bone, potassium, wood ash, calcium, lime, eggshells, pH of the soil affects the inorganic compounds of soil. So soil condition also having an exogenous influence factor in variation of plant size and the constituent present in them. Development age of herb, different components are produced in varying amount of different development stages. Vitamin C highest in rose hips just before maturity. This is rose hip fruits, Rosa rugosa. Camphor accumulate therefore collected from older trees. Camphor, the exudate, unorganized drug. Alkaloids highest in the young seeds, like caffeine which is present in the seeds of coffee. Young cloves, higher oil content, eugenol content is more than in the older ones. Mother clouds will be exhausted. Young buds will be having more eugenol, as shown in the picture. Herb, condition and parasites. Certain other factors also influence the quality of herb. Example, fungus or henbane lower the alkaloid content. Henbane is attacked by a fungus means the content of the alkaloid present in the tropane alkaloids will be less in henbane. It is therefore necessary to control pest and disease which may affect herbs and their medicinal value. Only possible when the herbs are cultivated. Use of growth promoting substances. Here comes the growth promoting hormones, cell division hormones, developing hormones, growth inhibiting hormones. So different types of hormones we have to study. Plant growth regulators, they are called as plant growth regulators, regulators can be defined as chemical substance that control all the aspects of growth and development within plants. They are also referred to as plant growth hormones or phytohormones. Plant growth hormones are organic compounds which are either produced naturally within the plants or are synthesized in laboratories. They profoundly control and modify the physiological processes like the growth, development and the movement of plants. Based on their action, plant growth regulators are broadly classified into two major groups: plant growth promoters, plant growth inhibitors. Auxins, gibberellins and cytokinins are groups into plant growth promoters, while abscisic acid and ethylene gas are grouped into plant growth inhibitors. Ethylene can be grouped either into the promoters or into the plant inhibitors. There is a small ethylene, they have action in two ways. So we cannot specifically ethylene can be added to growth inhibitor or growth developers. Plants growth hormones or plant growth regulators exhibit the following characters: differentiation and elongation of cells, formation of leaves, flowers and stem, wilting the leaves or wilting of leaves, falling down with less water content. Wilting is a natural response by the plant to reduce the water loss. They will reduce the water loss. This is done by the hormonal or the growth regulators. Ripening of fruit, seed dormancy, etc. Seed dormancy is the state in which seeds is unable to germinate even under ideal growing condition. Abscission is a natural occurring phenomenon which involves the separation of fruits, flowers or even leaves from plants at specialized separation layer. You can see the picture. Plant hormones applied for germination, flowering, maturity, ripening, seed dormancy, abscission. Types of plant growth regulators. The plant growth hormones or regulators are of following types: plant growth promoters, plant growth inhibitors, plant growth promoters. Auxins. Auxin, this is the structure. First starting with auxin. we can see that it is an indole derivative with a 6-membered benzene ring with a 5-membered ring with a nitrogen. We can say indole CH2COOH, abscisic acid, or sorry, not abscisic, this is auxins. The first phytohormone to be discovered is the auxin, and it was discovered by the biologist Charles Darwin. Auxin are one of the most important plant hormone. The chief naturally occurring auxin is indole 3 acetic acid, IAA, and other related compounds. The term auxin is derived from the Greek language meaning to grow. These plant growth regulators are generally produced at the point of stems and roots from where they are transported to other part of the plants. These plant hormones include both natural and synthetic sources. Indole 3 acetic acid and indole butyric acid are obtained from natural plant sources, whereas naphthalene, acetic acid and 2,4-dichlorophenoxyacetic acid are obtained from synthetic sources. Function of auxins: 1. To promote flowering in the plant, used to process of plant propagation, used to gardeners to keep lawns free from weeds, involved in the initiation of root in the stem cutting, prevent dropping of fruit and leaf in early stages, control xylem differentiation and help in cell division. Auxins are widely used in herbicide to kill dicot weeds, used for production of fruit without preceding fertilization, promote natural detached abscission or older leaves and fruits. Apial dominance may occur in which the growth of later bud is inhibited by the growth of apical bud. In such cases, the shoot caps may be removed. These are produced by the apex of the root and shoot. So these are the functions of auxins. Gibberellins are very important. This is the basic structure of gibberellic acid. It has a dicarboxylic acid group and a OH group. Gibberellins are an extensive chemical family based on an Ent-gibberellinae structure. The first gibberellin to be discovered as gibberellic acid. Now there are more than 100 types of gibberellins and are mainly obtained from a variety of organisms, from fungi to higher plants. They are acidic and are denoted as gibberellin A1, gibberellin A2, gibberellins A3, gibberellins A4, gibberellins A5, gibberellins A6, etc. There are hundreds of gibberellins have been identified now. This is a 6-membered ring structure. This is the 5-membered ring structure. No nitrogen is present. And this is 6-membered ring structure again. And there is again a 5-membered ring structure. These all the main 4 ring structures are present in gibberellins. One or twice if you write, it is possible for you to write. It is necessary for the students to write the structure, chemical structure of plant hormones and plant promoter or plant inhibitors or plant regulators. So OH present, and there is 2 dicarboxylic acid is also present in them. So this is the basic structure, and there is a CH3, that is the solid arrow, which is CH3 is also present in the basic gibberellins. And a double bond CH2 and ethylene is present in outside the ring structure. Is a special character for gibberellins. So this is the basic and gibberellin A numbering from 1, 2, 3, starting from the 1, 2, 3, 4. Fourth position, there is the methyl group and the acidic group will be there. And in the second position also OH group will be there. For example, you can see third position there is an OH. When you see what is the difference of G1, GA1, the double bond is added from the GA1. Once you write the GA1 structure, the student have to study like this. Double bond is added and it becomes GA3. And here OH is removed from the original structure. The structure is just tilted and OH is removed. So that becomes GA4. And here you can see OH is removed and also double bond is added. So that is GA7. And GA6 you can see OH is removed from the other side and a 3-membered lactone ring as shown in the structure. And here in the GA5 OH is removed, a double bond is fixed in the, which is dealing with the 3 and 2 having a double bond, not in the 2 and 1 or 1 and 2. This is 2 and 3, there is a double bond in the structure. So this is how we can write the structure, and you can study at least this 6 structure which I told. Functions of gibberellins: they delay senescence in the fruit, involved in leaf expansion, break dug and seed dormancy, promote bolting in cabbage and beetroot or beet, and help fruits like apples and elongate and improve their shapes. Used by the brewing industry for increasing the speed on the malting process. Used as the spraying agent to increase sugarcane yield by lengthening of the stem. Used to fasten the maturity period in young conifers, cone-shaped seedlings, conifers, and promote like corn early seed production. Helps in increasing the crop yield and increase the height of the plant, such as sugarcane, and increase the axis length in the plant such as grape stalks. Gibberellins are acidic in nature. This is about the cytokinin, cell division hormone, where there is an, you can see that is an arrow which I am rounding, which shows that it is a purine derivative with NH2 and a side chain NH2, CH2, CH double bond C, CH3, CH2, OH. And these are produced in the regions where cell division occurs, mostly in the roots and shoots. They help in production of new leaves, lateral shoot growth, chloroplast in the leaves, etc. They help in overcoming apical dominance and delay aging of leaves. Function of cytokinins: they break bud and seed dormancy, promotes the growth of the lateral bud, promotes cell division and apical dominance. They are used to keep flower fresh for a long time, used in tissue culture to induce cell division in mature tissues, promote lateral shoot growth An advantageous root formation helps in delaying the process of aging, senescence in fresh leaf crops like cabbage and lettuce. Involved in the formation of new leaves and chloroplast organelles within the plant cell, used in induce the development of shoot and root along with oxygen, depending on the ratio. Plant growth inhibitor, abscisic acid. This is a side chain which we can see that very clearly. That is CH double bond CH, C, CH3 double bond CH and C double bond, both that is COOH acid group, and then a substitution with a double bond with CH3 and a ring structure with a double bond O. It's a structure of quinone, what we can say. So abscisic acid, it's a growth inhibitor which was discovered in 1960s. It was initially called dormant. Later, another compound, Abscission 2, was discovered and are commonly called as abscisic acid. This growth inhibitor is synthesized within the stem, leaves, fruits and seeds of the plant. Abscisic acid mostly acts as an antagonist for or antagonist to gibberellic acid, which is a growth hormone, development of the stem. It is also known as a stress hormone, as it helps by increasing the tolerance of the plant to different kinds of stress. Function of abscisic acid: stimulate closing of stomata in the epidermis, helps in development of maturation of seeds, inhibits plant metabolism and seed germination. It is involved in regulating abscission and dormancy. It is widely used as a spraying agent on trees to regulate dropping of fruits, induces dormancy in seed and helps in withstanding desiccation and other unfavorable growth factors. Ethylene, CH double bond CH2 gas. Ethylene is a simple gas plant growth regulator synthesized by most of the plant organ, including ripening fruits and aging tissues. It is an unsaturated hydrocarbon having double covalent bond between the adjacent of carbon atoms. Ethylene is used as both plant growth promoters and plant growth inhibitors. Ethylene is synthesized by ripening fruits and aging tissues. The gas which is evolved. Even there is a saying that when you keep oranges with bananas, banana will get ripe very fast because of the gas which is produced from the skin of the oranges. Functions of ethylene. Ethylene is most widely used plant growth regulators as it helps in regulating and physiological process, induces flowering in mango tree, promotes sprouting of potato tubers, break the dormancy of seed and bud, enhances respiration rate during ripening of fruits, applied to rubber trees to stimulate the flow of latex, promotes abscission and senescence of both leaves and flowers, used to stimulate the ripening of fruits, for example tomatoes and citric acid, affects the horizontal growth of seedlings and swelling of axis in dicot seedlings, increase root growth and root hair formation, therefore helping plants in increasing their absorption surface area. Allelopathy, this is an important in short notes definition. In MCQ they may ask about allelopathy. Define the constant of effect which living organism exerts on each other, which may be either beneficial or harmful. This is a definition for allelopathy. When it is attached to another person, A and B, may be beneficial, may be harmful. In plants, different plants growing together affect factors such as germination rate, leaf development, fruit maturation, chemical constituent produced. Allelopathy affects transmission between plants in a number of ways. Exaltation of leaves, root secretions, extractions from falling leaves into the soil, mutual development organism benefits, which is called as symbiosis. Symbiosis means something benefited if A and B together. A is benefited by B or B is benefited by A, then it is symbiosis. One example of symbiosis is relationship between certain species of ants. It's interesting, ants in acacia trees. The ants live in the hollow of the thorns and of the acacia tree as shown in the picture, and helps to protect the tree from various other herbivorous animals or the herbivores and to attack or to eat the leaves or the bark. Marigold are essential for your garden, very important. They are also an example for symbiosis. In several ways, as they are not only looks very nice or tinge or beauty in your garden, they helps the other plants in their surroundings in the garden. You can see the fly which is there, that is hoverflies, not fly, hoverflies. This symbiotic plant produces a flavors, scent that attracts the hoverflies. These hoverflies feed off green fleas, plant lice and twitch grasses. They are weeds. They will affect other flowers, other plants. So this will be protecting. This will act as a natural pesticide. Like top benefit of marigold in your garden. Destruction allelopathy, which is negative, then it is called antibiosis. Antibiosis. So symbiosis, antibiosis. Short notes. Allelopathy. Clearly a student can write with example. Example for antibiosis, belladonna growth is inhibited when it is cultivated next to a mustard plant. If you are growing belladonna plant with mustard plant, it is getting affected, inhibited, the growth is inhibited. Finished. One example for antibiosis. Other factors affect the exogenous factors in the variation in the plant, affecting the plant growth and isolation differently than the plant growing community. Example, camphor tree produces camphor when growing alone than those growing in groups. Ergot is an actually by bacteria. For example, alkaloid content differs, ergometrin, ergotamine. All this content differs according to the host, ripe species and type of cereals like barley or oats. Variation in actives or active constituents. Example, foxglove digitalis produces more active constituents during the day than at night. Now we are coming to the second plant variation factor number 2, which is endogenous, inside the plant itself, genetic factors. So what is there? Endogenous factors, genetic differences are responsible for development of the new plant crop variation, plant genetic resources and plant genetic diversity, morphological variety, biochemical diversity among type of chemical produced, chemical races. So we should know about, even short notes they have asked last time, polyploidy, hybridization. So the students should write very clearly, not like A and B together giving C is hybridization. No, you should write clearly. Polyploidy means somatic number of chromosomes present in the plant cell nucleus. Normally, the same of all tissues of the same plant normally are even number, possibly to divide the chromosome into pairs. So haploid, single, diploid, triploid and tetraploid and polyploid is also, each number of pairs are identical. Term diploid, in some plants chromosomes are not equal to diploid, so triploid, tetraploid, octoploid, polyploid plants. So when you see polyploids in the picture, you can see polyploidation, giga effects, which gives, when you see polyploid, see the plant shoot system, how the changes, how the grafting from the stem when they are doing. So natural occurring polyploid, mentha, common crops like wheat, oats, cotton, artificially induced by heat treated, addition of specific chemical agents can also, polyploidy can be done. Effects of polyploidy, varieties of changes, plant size, organ size. In the picture we can show that shoot system changes, individual cell size, change in the chemical constituent production. Effect of polyploidy beneficial, example, deadly nightshade, Atropa belladonna, double the, that is X square alkaloids than an diploid when you do polyploidy. And when you do a papaver somniferum, presence of opium poppy, latex is X square morphine than a diploid plant. Carum caravi, that is caraway, volatile oils, yields more than the diploid. So diploid constituents also having increased when it is becoming a polyploid. Non-beneficial potential, reduced growth, reduced plant vigor and reduced plant fertility. Plant breeding methods influence morphological and chemical variety in plants aimed at reducing variability of these factors naturally produced by genetic differences. At producing plants with constant desirable characters, 3 methods: selective breeding, hybridization, transgenic medicinal plants. These are all short questions the student should know. Selective breeding means what? Individual plants showing the most desirable characters are shown or chosen and interbreeded to population. Improved quality. Reason, when a single species of plant appearing individually or identically, when a single species of plant appearing identical, genetic variability are present making them slightly different from each other. Results, improved growth rate, disease resistant, winter hardness resistant and increased yield of medicinal activity. So for an original Brassica plant, when you are doing this grafting or a selected, you can see selected for a stem, Cohira B is obtained, selected from a lateral bud, Brussels sprouts you can produce, selection of terminal buds, cabbage, selection of flower cluster you will get cauliflower, and selection of a stem and flower broccoli, selection for leaves kali. So one plant example like brassica plants can be selective breedings can be done. Hybridization of the plant, parent line A like a flower, parent line B, and the color is completely different from red and blue, it becomes an maroon in F1 hybrid. So mating of different species or variety to produce a hybrid pregnancy different from both parent, incorporating desired characteristic as shown in the picture. Example, Mentha purpurata and Mentha spicata are natural occurring hybrid. These pictures which I have shown, Mentha acutica, Mentha longifolia, Mentha spicata, Mentha rotundifolia, Mentha arvenicis, and this is Mentha purpurata. So there are many, but these two species are hybrids naturally. Transgenic medicinal plants. What is this transgenic medicinal plants? Genetic engineering, coding, recombinant DNA, transfer DNA sequence from chromosome of one plant to another, leading to an artificial transfer of a particular character of one organism to another. So you see, plant expression constitutes a recombinant vector, and you can produce a transformation of transgenic plant of vitro culture, and biologically active compound extraction can be done, which can be produced to anti-cancer drug, antioxidant, antifungal, antibacterial, antiprotective, and neuroprotective drugs can also be produced, or any vaccines can also be produced. These are some of the examples of transgenic plants such as tobacco, banana, algae and potato are known to produce vaccines and antibodies against the gastrointestinal tract disorders such as cholera. And pharmaceutical products range from recombinant hepatitis B vaccine produced by gene modified Baker's yeast. GM means gene modified, coding modified, adjusted, recombinant DNA, gene modified Baker yeast to injectable insulin for diabetes. Insulin can be produced in a gene modified E. coli, Escherichia coli bacteria, which can produce insulin for diabetes. Plant propagation. Seed, example, digitalis. The seeds of digitalis prior to sowing may be influence the germination. Eggplant needs soaking before germination in water and acid or dilute acid. Vegetatively, example, bulb, tubers, rhizomes. By division, plants separate from aerial parts, buds such as roots, growing points. Runners, example, mint. Stolons, example, licorice. Cuttings can also be done, grafting, fermentation. Inoculation can also be done vegetatively. Seeds, plant propagation can be seeds, and vegetatively can also be done by grafting, fermentation. Inoculations can also be done. Cell culture, because we have a chapter of tissue culture, a big one more chapter, one unit. So in that we will discuss about the tissue culture. So that is also plant propagation. And now the third one, the plant variation factor number 3, how it is influencing, there is post cultivation factors. Harvesting of herbs, roots and rhizome collected when aerial parts are dried down, they will find out it is mature. If not, freshly dried to dry. And When you come to bark, damp weather, aerial part at a flowering, that is active photosynthesis, and flowers like clove before fully expanded. Fruits vary, some of them ripe, fully ripe anise fennel, nearly ripe cardamom, before the seeds should be dispersed, dispersal stage, gums and resin dry weather. Preparation of herbs: remove the sandstones, wash under parts, remove rootlets, diseases portion should be sliced, and larger organs, peeling off ginger, licorice, removal Naxomica, and packing in the squill for cinnamon, and grading like senna and gums. Drying of crude drugs: aim to remove the excess moisture, ensure good keeping, and prevent mold, bacteria growth, and prevent enzyme activity, preventing chemical changes. Ideal herbs should be dried as soon as possible. ASAP means as soon as possible, for collection, maintenance, appearance, and chemical activity. Drying methods like air drying, artificial drying, vacuum drying, the drying techniques whichever you have studied in a pharmaceutical engineering can be applied. Air drying, simply sun drying, when herbs are not adversely affected by the excessive sunlight, dried in thin layers. Then occasionally it can be turned occasionally, like clove and cardamom. And shade drying, when sunlight causes decoloration, wrapping, swirling like cinnamon can be done in shade drying. So these are some of the drying techniques which can be done manually. And artificial drying, generally the most acceptable form of drying herb: rapid loss exposure to heat, less change in chemical alteration. Control temperature, normally 40 degrees centigrade. Ventilation is needed, allowed dry air to replace wet air. Vacuum drying, steam heat ovens, pump used to extract air, low pressure to ensure rapid and complete drying. Expensive method, reserved for expensive herb and which cannot be successfully dried through other methods. Generally, drying techniques of leaves, we have many leaves. Leaves are dried in 40 to 50 degrees centigrade, maintain good color. Without over drying herbs, normally destalked first, stalks will be removed. Just drying short period of time only prevents swelling, discoloration. Leaves may be bleached through the drying process. Best drying in dark. Drying techniques for roots and rhizome. You may have many rhizomes which we are coming across, ginger and many other. Rhubarb, washed, sliced, dried between 30 to 60 degrees centigrade. Temperature too high, form a touch crush on the outside, preventing the surface drying deeper tissues. Thick organs may require long period of drying, 10 days to 3 weeks. Avoid mold growth. Generally, drying technique for flowers. We have many flowers also for volatile substances. Normally, stalked before drying. Flowers may also bleached when dried. Best drying in dark. Generally, drying techniques: volatile oil containing herbs, air dry at low temperature. Avoid volatilization of volatile oil content. Should be dried separately with other herbs. And general drying techniques for fruits and seeds. Normally, partially dried before harvesting, maybe air dry afterwards. Seeds are normally separated from the fruits before drying. Fruits are normally left whole. Some fruits, cardamom, ruptured due to excessive heat. General drying for bark, occasionally drying and removal of outer layer. Which drying technique to choose is also depending on the physical character of the herb, whether it is a leaf, flower or bark or rhizome. According to that, chemical character of the herb, like volatile oil or starchy herb. Volatile oil means separately low temperature, starchy means gentilized and not overheating, and alkaloids means becoming deactivated if the dried excessive high temperature, like opium, poppy. So change occurring in the herbs when drying are changes due to enzyme action, oxidation, volatilization and browning of the drug can be happen. Adulteration of the crude drug also comes after processing, intentionally, non-intentionally, deliberately, undeliberately, like that I have detail given in the adulteration classes. But this also should be added. It is also a point of and factor after processing can also affect the variation in the plant quality and its constituents. So adulteration of the herb does not conform the official standard. Usually practice when the herbs scarce or expensive, then they will go on doing this adulteration with inferiority, spoilage, admixture, sophistication, deterioration, substitution. These words they will ask usually the definition. Inferior herb quality leads to substandard herb, results naturally in nature, ignorance, carelessness, collection of genuine material regardless of the time, factor, collecting herbs prior, after the ideal time. That is the example. Collection in incorrect stage of development. An example, coriander full ripe, fruits should be nearly ripe. Clove, the flowers should be young and the bud should be collected. Collection of parts not medicinally valuable. Example, chamomile leaves. And collection of incorrect herb resembles the same herb also, one type of inferior herb quality. Imperfect preparation, not removing undesirable part and which is already present. Cork with the ginger rhizome, incorrect drying condition, these all are inferior herb quality. And incorrect storage, volatile oils cool, dry placed in a dry airtight container. So that is also one type of concept. Then herb spoilage means getting spoilage. You can see the picture, the leaves, how it is spoilage. When the quality of the medicinal value, usefulness of the herb is impaired or destroyed. Bacterial, fungal action, insects, rodents. Normally results in incorrect storage, affects the water and temperature, will affect this mold development. Herbs which have been contaminated by bacteria, fungus growth should be rejected. Deterioration means this also freeze damage, delay, carbon dioxide injury. We can see the picture how decayed an injury is taking place, freeze damage. So value of quality of the herb has been impaired. Extraction, destruction of the medicinal activities occurring commonly when the herb is expensive, limited or needed of numerous purpose. Example, ginger, removal of medicinal activity, constituents in making ginger beer, cooking purpose. Aging, heat, volatile oils, moisture and light, oxidation promotes the destruction of vitamin A in some of the compounds. Admixture, when a herbs or another species are added to medicinally valued herb, that is called admixture. Maybe intentionally or resulting collection due to unskilled labor also. Presence of plant parts other than those allowed. Excessive stock present in the flower or the leaf. Collection of foreign material, stones, dirt. Certain herbs may be legally mixed with the inert and other materials. Sophistication means addition of inferior material which are intended to decrease the medicinal action of the herb. Example, candle wax colored into yellow and being offered as a beeswax. Example, addition of flour powder in a ginger with a chilies for potency. And substitution means addition of allied drug or one which is botanically different, like digitalis thuspi with digitalis purpurea. So this is the final slide which you want to say, that is factors creating adulteration in authentic crude drug. So when you want to authentic a crude drug, this when you are talking about adulteration, sophistication drug, inferiority drug, admixture drug, and complete substitution or spoilage or deterioration drug. When it comes to, it should be authenticated, and impaired quality should be identified the adulteration, and then standard or adulterated drug can be determined by the pharmacist. So these are the factors creating adulteration in authenticating crude drug. So when we have seen about the cultivation collection in three sectors, like exogenous factors, endogenous factors, and factors after cultivation and after collecting, after processing of the drugs which is collected after cultivating or after starting from harvesting. So this also included. Thank you very much. All students, you just listen and see these notes, and so that it will be easy for you to attend 10 mark question, 5 mark question, and also for 2 mark questions, and also for MCQs in various government exams and any other exams. Cultivation of medicinal plants, very short form I have explained. I have made this very short, and the definition, the names whichever we have discussed in the first slide itself, we have discussed all the definitions which were told in the beginning. Thank you for listening to my class today.